Science is awesome and I love it so. There's still lots and lots of things science don't know. There have been major discoveries throughout history, but this universe of ours is still one great big mystery. It'll be perfect and beautiful, it'll be testable and provable, and that's what I like. So science is awesome and I love it so. There's still lots and lots of things science don't know. There have been major discoveries throughout history, but this universe of ours is still one great big mystery.
Welcome to Bacteria. Today's lecture we are going to talk about the exchange with the environment. The goals of this video are essential question: how do bacterial cells work to carry out the 5 characteristics of life?
Specifically, we're explaining how molecules are transported in and out of the cell and the difference between active and passive transport.
When we are talking about how molecules are transported in and out of the cell, we know that we have a cell membrane surrounding every cell. The cell membrane of bacterial cells control everything that comes in and out of the cell. Material either enters the cell through the membrane, through the membrane proteins, or right through the phospholipid bilayer.
Sometimes a cell needs to use energy to move things in and out of the cell, so it can't simply move right through the membrane. The cell actually needs to actively transport it through channel proteins, and this is called active transport.
Other times, the cell does not need to use energy to move materials in and out of the cell, and this is called passive transport.
When we're talking about passive transport, there are 3 words we use to describe this. We will use diffusion, osmosis, and facilitated diffusion. Passive transport only works when material is moving from where there is more of the material to where there is less of the material. And we call this moving from a high concentration to a low concentration, or we call it moving down a concentration gradient. Alright, so here you can see a cell membrane in the middle, and on the left there are a lot of the red dots, a lot of the red material, and diffusion is happening because some of the red dots are moving over to the right where there was a low concentration. And at the end, it kind of evens out. Hey guys, we have a piece of dialysis tubing with a really high concentration of salt dissolved in water. Inside, we put some red food coloring, pink food coloring, so you can see what's going to happen. And then in this large beaker, we have just pure water, so water with nothing dissolved in it. In the tube, again, we have water with a lot of salt dissolved in it and a little bit of food coloring added. We're going to put this in the water and let it sit for a while and see what happens.
So now we're going to talk about concentration and solutions. And a solution is a mixture of a substance dissolved in water. And in a solution you have 2 parts. You have a solvent, and that's your water particles, and you have a solute. That's the thing you're dissolving in the water. So concentration describes how much of the solution is water and how much of the solution is the dissolved particles. For example, if you make a mixture of salt water, and you have your water, which contains 80% of the solution, and your salt contains 20% of the solution. Here is an example of a solution. Our solvent is the water, and you can see we have little solute particles. In the example that we used, those solute particles would be salt. So this solution from our example would be 80% water and 20% salt. So the concentration of salt is 20%, the concentration of water would be 80%.
So in this picture you can see a gradient of concentration. So the cups on the right are more concentrated than the cups on the left. When things are less concentrated, we call them diluted. The animation on the bottom just shows what's going on inside those cups. So the blue dots represent water molecules, and the red dots represent the dye that's used to dye the cups red. So you can see at the bottom in the animation that the cups on the left don't have as many red dots as the cups on the right. So we say that the cups on the right have a higher concentration of dye than the cups on the left.
So sometimes we need to compare solutions, and remember that the cytoplasm of any cell is a solution. It's water with things dissolved in it, and all cells are surrounded by solutions. So when the solution surrounding your cell has a lower concentration of solute and a higher concentration of water than inside the cell, we call it hypotonic. So that means the solution outside of the cell has a higher concentration of water and a lower concentration of solute. So in the picture on the left, we can see that red cell membrane, and inside the cell it's 20% salt, which means 80% water, and outside the cell it's 10% salt, which means 90% water. So inside the cell there's a higher concentration of solute and a lower concentration of water. So as we saw in the animation earlier, concentrations try to even themselves out, so things will move from high concentration to low concentration. So since the concentration of water outside the cell was higher, it will move inside the cell to equal it out. The cell will expand and get a little bit bigger, and so one way to remember this is it'll get big like a hippo, and so that can help you remember hypotonic solution. Hippo.
There's also another solution called a hypertonic solution. And in a hypertonic solution, the solution has a higher concentration of solutes and a lower concentration of water than inside the cell. So you can see from our image down there, inside the cell there's 10% salt, so that means there's 90% water. And the solution surrounding the cell, there's 20% salt, so that means there is 80% water. And water wants to go from a high concentration to a low concentration, so it's going to diffuse out of the cell because in the cell there's 90% water, and outside the cell there's 80% water, so it's going from high to low. When we talk about water diffusing out of a cell, we call it osmosis. So osmosis is diffusion of water. One way to remember this, a hypertonic solution, you can think of hyper, very active, and being very skinny like that little boy. So what's going to happen to the cell is going to shrivel up and shrink and look really skinny just like that little boy flexing.
So in this third picture we can see that inside of the cell membrane, the red cell membrane, there is 10% salt, which means 90% water, and outside of the cell membrane there's also 10% salt, so 90% water. In this case the concentration of water is equal to each other. We call this isotonic. So in isotonic situations, the amount of water going into a cell is going to equal the amount of water going out of a cell. Your cell is trying to maintain an isotonic situation. This is just part of keeping homeostasis inside the cell, keeping a constant environment.
When we talk about this movement of materials from a high to low concentration down a concentration gradient, we call it diffusion. The diffusion of water is called osmosis. Water and oxygen are two very common substances that a cell will move across its membrane using passive transport with no energy.
So when material wants to get inside of the cell, the material can go either through the phospholipid bilayer or it can go through the proteins embedded in the phospholipid bilayer. If the material or particles are going right through the phospholipid bilayer and not using the membrane proteins, it's called diffusion. But when material moves across the cell and it uses the membrane proteins to get into the cell, it's called facilitated diffusion.
There's other times where the cell needs to move material against the concentration gradient. In these situations, there's more of the material inside the cell than outside the cell. In these situations, the cell needs to use energy in order to move materials against the concentration gradient to where there's more of the material. When a cell uses energy to move materials, it's called active transport. Embedded in the cell membrane, there's special proteins embedded in the phospholipid bilayer that move materials across the cell membrane using energy, using active transport. Usually there's specific proteins for specific materials that have to move against this concentration gradient. So the picture at the bottom illustrates this going from a low concentration to a high concentration. So we can think of it as a person on rollerblades or roller skates going up a hill. So they're going from a low point to a high point. So they have to use a lot of energy, to do a lot of work in order to go against this, to go from low to high.
So here you can see some pictures of all of these different types of transport. All the way to the left we have simple diffusion, passive transport right through the lipid bilayer, high concentration outside the cell, low concentration inside, so the material is moving inside the cell. Letter B is an example of facilitated diffusion, so we have a non-specific transport or a protein that will just allow any material inside the cell. In letter C, we have active transport through a specific transporter, so glucose in this case needs to get in the cell. The cell will use energy to bring glucose into the cell. And in letter D, we have osmosis two ways. On the left, it goes right through the lipid bilayer, and in the center of the picture of the membrane, water is going through another one of those specialized proteins that will help materials get into the cell.
So to recap, our essential question was, how do bacterial cells work to carry out the 5 characteristics of life? Specifically, we wanted to look at how molecules are transported in and out of the cell. And we talked about 2 different types of transport, passive transport and active transport. And for passive transport, we talked about diffusion, osmosis, and facilitated diffusion. And for active transport, we talked about molecules getting in and out of the cell using special membrane proteins that require energy. And these molecules are going from a low concentration to a high concentration.
And remember, the difference between active and passive transport is that in active transport, the cell is using energy to move materials against a concentration gradient from low concentration to high concentration. In passive transport, there's no energy that needs to be used because the cell can just use the concentration gradient going from high concentration to low concentration. And that is all.
All right, let's see if we can get that salt video in.